Hello everyone. Today we're going to talk about the different types of channel proteins and carrier proteins. Certain molecules can pass directly into or out of the cell directly via the plasma membrane, but there are some molecules that are either too big or they carry charges like ions, and those cannot pass directly through the plasma membrane. So those would need to pass through what's known as a channel protein. Channel proteins, therefore, can allow the transport of ions. They can also allow the transport of water. And water actually has the ability to either pass directly through the plasma membrane or it would pass through one of these channels. Now, the diameter of the channel protein plays a role in which substrates are going to be allowed to pass through it. Also, the charge within that channel protein plays a role in determining which substrates will pass through it. Some of these proteins are known as open channels, or we refer to them as leaky channels. Those leaky channels are actually open most of the time, allowing substrates to either pass into or out of the cell. Some of those channels are known as gated channels, and the reason why they're known as gated channels because most of the time they are closed, and they will only open based on certain circumstances. So some of these gated channels, we refer to them as chemically gated channels because they will only open if the correct chemical or the correct substrate binds to that channel, forcing it to open. Some of them are known as voltage-gated channels, and those voltage-gated channels are only going to open if the electrical state inside of the cell, let's say, becomes more negative, or if it becomes more positive. So if the electrical state inside of the cell varies, that's what's going to trigger those gated channels to open. Some of them are mechanically gated, and so those are only going to open if, let's say, the temperature increases or if the pressure inside of the cell increases. Now, carrier proteins, those proteins, they are never going to be opened to both sides. So let's say this is outside the cell, facing the extracellular fluid, and let's say this is the side facing the inside of the cell or the intracellular fluid. Notice how the carrier proteins are always going to be closed at one end versus the channel proteins. Once they open, they will be open to both sides. Now, those carrier proteins, for them to face or open towards the other side, they have to change their conformation, like they have to change their shape. And so therefore, carrier proteins are not going to allow the transport of molecules as fast as the channel proteins. They're much slower due to that conformation change that they have to go through. Some of these carrier proteins are known as uniport carrier proteins because they only allow one substrate to pass through them. Whether this molecule is going to be passing into or out of the cell, as long as it's one substrate passing through, it's known as a uniport. Symport carrier proteins allow two or more substrates to pass through, and those two substrates are moving in the same direction. So those two substrates are either entering the cell or they are exiting the cell. Antiport carrier proteins allow two or more substrates to pass into or out of the cell, and they allow those substrates to move in opposite directions. So as you can see in this image right here, the sodium is exiting the cell while the potassium is entering the cell. So that's known as an antiport carrier protein. This image right here shows how a carrier protein goes ahead and transports a molecule into or out of the cell. So the molecule that needs to be transported, we can refer to it as a substrate, or you can refer to it as a ligand. It has to fit into a special site on that carrier protein. Once it binds to that carrier protein, The carrier protein is going to go through a conformational change where it's going to go ahead and close that side that was already open, and it's going to go ahead and open on the other side, allowing the transport of the molecule. Now, a carrier protein that allows glucose to pass through, we refer to it as a glucose transporter, or GLUT for glucose. So that glucose transporter allows the transport of the glucose molecules. And as you can see from this image right here, it's facilitated diffusion. Facilitated meaning help. So those glucose are using the help of those carrier proteins to basically pass through the cell. Facilitated diffusion is a passive transport, meaning that it does not require any energy. And the reason why it does not require any energy, because as you can see, the glucose molecules are moving from the high concentrated area to the low concentrated area. Whenever a molecule is moving from the high concentration area to a lower concentrated area, that's known as a passive transport, and no energy is going to be required. Now, on the other hand, active transport are known to need energy to move those substrates into or out of the cell. And the reason why active transport needs energy, because those molecules are going to be moving against their concentration gradient, meaning that those molecules are going to be moving from the low concentrated sides to the high concentrated sides. And that means that those molecules are basically going against the flow or against that concentration gradient, and so therefore it would need energy. Now, active transport usually uses carrier proteins to allow the transport of these molecules. Some of these active transports are going to use ATP energy directly to allow the transport of these molecules. And so we refer to those carrier proteins as primary active transports. Some of them, we refer to them as a secondary active transport, and that means they're not going to use energy directly, but the way they are going to move the substrates is that they are going to rely on the kinetic energy or the potential energy of one substrate to allow the movement of another substrate. I'll show you an example to make it more understandable. But let me show you this table right here. This is showing you a bunch of primary active transports. So those are carrier proteins that allow the passage of substrates against their concentration gradient. And to allow the transport of these substrates, ATP is used directly. So the one that we are going to talk about is a carrier protein known as the sodium-potassium ATPase, or you can refer to it as the sodium-potassium pump. It's an antiport, meaning that sodium and potassium are going to be moving in opposite direction. If we take a look at our intracellular fluid and our extracellular fluid, we would find that we have a higher concentration of sodium ions and chloride ions outside our cell or in our extracellular fluid compared to inside the cell or our intracellular fluid. Our intracellular fluid has a higher concentration of potassium ions compared to outside the cell or in the extracellular fluid. So an active transport is going to allow ions to move against their concentration gradient, basically from a low concentrated side to the high concentrated side. So where do we have much lower sodium ions? In our intracellular fluid. So the sodium ions are actually going to be exiting the cell. Same with the chloride ions. They're actually going to be moving towards the extracellular fluid because sodium and chloride are moving from the low concentrated side to the high concentrated side. That's an active transport, and it's going to require ATP. Now, potassium, how's potassium going to move? Potassium is actually going to start entering into the cell because we actually have more potassium inside of the cell. And so you would notice that the sodium and potassium ions, they're moving against the flow. And if they are going to move against the flow, they need to pass through a carrier protein known as the sodium-potassium pump, and they are going to use ATP directly to help them with that movement. This image right here shows you exactly how sodium and potassium are going to move against their flow using that sodium-potassium ATPase. So here's the carrier protein right there. And you can see that this carrier protein has special sites where 3 sodium ions are going to go ahead and attach to the carrier protein. So 3 sodium ions are going to latch on to that carrier protein. And as I said, ATP is going to be used directly. So ATP stands for adenosine triphosphate. So tri meaning that it has 3 phosphates. ATP breaks down, or we use the term hydrolyzes, and when it breaks down, you get ADP, 2 phosphates, diphosphate, and you get an extra, or not an extra, you get that third phosphate on its own. That third phosphate binds to the carrier protein, and when that phosphate binds to the carrier protein is going to go through a conformational change. And it's going to go ahead and open to the other side, and it's going to allow the transport of these sodium ions outside of the cell. So it's going to move 3 sodium ions into the extracellular fluid. Now, sodium-potassium ATPase, it's an antiport. So it's going to move 2 substrates in opposite direction. So 3 sodiums are going to move out, and then what happens? 2 potassium are going to bind, and they're actually going to be transported into the cell. So 2 sodium ions are going to go ahead and bind to that carrier protein, where it has to go through another conformational change, and it's going to open towards the inside of the cell, moving the 2 potassium ions inside. And that's how the sodium-potassium pump works. 3 sodium ions move towards the outside of the cell, and 2 potassium ions will move inside of the cell. And to help with that movement, ATP is directly used. Now, secondary active transporters, they're also going to allow the substrates to move against their concentration gradient, but this time they're not going to use ATP directly. The one that we are going to talk about is the sodium glucose transporter. The sodium glucose cotransport, it's an active transport. The way it works is that it's going to use the kinetic energy of one molecule moving down its concentration gradient to push another molecule against its concentration gradient. So in this case, sodium is actually moving down its concentration gradient. What does that mean? It's moving down its concentration gradient. That means that sodium is moving from a high concentration area to a low concentration area, and that's a passive transport. But when sodium goes ahead and binds to that carrier protein, what happens is, think of it as sodium kind of doing all the work. And so when sodium goes ahead and latches on to that carrier protein, it's going to create an active site for glucose to open up. And so glucose, think of it as it's getting a free ride. So once the sodium latches on, glucose quickly binds to the carrier protein at the same time. And so the sodium moves from the high concentrated area to the low concentrated area. But the way glucose is moving, it's actually going from the low concentrated area to the high concentrated area. And so the sodium glucose transport moves sodium down its concentration gradient while it moves the glucose against its concentration gradient. And so glucose is moving against the concentration gradient, not via the help of ATP, but via the help of the sodium. So only because sodium latched on first and it moved from high to low, that's what allowed the glucose to move from low to high.